second step is target validation. Target validation means when the drug is now when the target is identified, now the researchers, they want to confirm that whether this is a target which is actually responsible for the development of the disease or not. So that target is then validated, and for validation of the target there are various processes which are used, and generally the target validation is conducted in animals and in the transgenic animals. So there is a process by which the target is validated. Then after that is hit discovery. Hit discovery means now there are huge chemical libraries and databases which are there with the researchers. They are with the research institute. They have there are various chemical databases, and there are various huge libraries which contains lakhs of chemical molecules. Some are having and all are having some or the other activity against the disease. Some are having the activity, but they are very toxic. Some are not toxic, but they have very less activity. Some are not active at all. Some are active also and effective also, but they are not binding with the receptor, or their binding with the receptor is very weak, and they dissociate with the receptor very easily or very fastly. So now we have to discover. So now again, from those chemical libraries, there are various computational methods which are used, which is known as the process of computer aided drug designing. And these processes are known as virtual screening, molecular docking, molecular modeling. So these all processes are used, and by the drug, when it undergoes all these processes, we come up with certain chemical molecules which have some activity for the disease, against that disease. Means they can bind with the receptor. They have good scoring functions to bind with that receptor. So we will consider only those molecules for further assay development and screening, because then after in laboratory also, in the cell cultures, the drug is tested to see the effectiveness of the drug and to see the affinity of the drug to bind with the receptor. So the drug should bind with the receptor with highest affinity, with the selectivity and specificity. Means a drug should bind with a particular receptor only. Okay, so the drug should have the selectivity to bind with that receptor. Okay, so that all procedures are done. Then how high throughput screening? So high throughput screening was an old method which is not used now. Virtual screening, which is a new method in which various the softwares are used. So virtual screening and molecular docking method, which is used for screening the compounds which are having the maximum activity or maximum affinity for the receptor. And then if they are binding with the receptor, then only we will screen them for its efficacy and safety. Then after that, the compound which is hit to lead, means we will in this process we'll get a chemical compound or a ligand which has all the properties, all the characteristics, that it binds with the receptor effectively, it binds with the receptor selectively and specifically, it is having the effect, it is effective against the disease, and it is also safe. But now that all safety and effectiveness we have got from the cell cultures from in the laboratory, now we have to see that what is the actual effect of the drug. So for that we need to conduct the drug has to undergo the drug development process. And then after that, hit to lead, that is lead identification. That process is also known as lead identification. And then the next process is lead optimization. So lead optimization, in which we try to understand that what is the physiochemical properties of the drug, like what is the ionization, what is the partition coefficient, how the drug is bind, and all these properties are very useful to understand that how to get the knowledge about the stability of the drug, how the drug is binding. FND studies are conducted, that is formulation development studies are conducted to decide that in which formulation the drug is stable, in which formulation it will have the maximum therapeutic action. Okay, so all these studies are done. And then and what will be the method of synthesis, because for the method of synthesis also there are various methods which are followed. So by which method we are getting the maximum yield and which is also cost effective. So all these processes are done. So that process is known as, so that all is done during the lead optimization study. And after that, the formulation and development studies, manufacturing, scale up studies are done. Then after that, the drug is now ready to introduce into the drug development process. And then after that, the drug development process starts. So drug development process starts from here, when the drug is tested on animals, and it is known as preclinical studies or non-clinical studies. Okay, so now these studies are conducted in vivo, in vitro, and ex vivo. So do you know what is in vivo, in vitro, and ex vivo? Any part of living organism, it is taken out generally, and that is which is known as bioassay also. Okay, on any test which is performed on human biomaterial, okay, on living biomaterial, that is on any tissues, generally it is performed on the intestines. So that is known as ex vivo. Okay, so various in vivo, ex vivo, and in vitro assays are conducted. So now, before discussing about the preclinical studies, I just want to tell you about the pharmacological processes. So do you know what is pharmacology? Simple, what is pharmacology?
It is the action of the drug. It is a study, basically it is a study of the drug on the body. Yes. And action of the drugs on the body.
Of
and
course,
what
correct.
The body does.
Simply, if you want to define pharmacology, so you can define as that pharmacology means it is a study of drugs. So we study about the drugs, like how it is behaving when it is going inside the body, it is administered to the body, what is the mechanism of action. So now there are two processes which we generally study in pharmacology, that is pharmacokinetics and pharmacodynamics. So what is pharmacokinetics and pharmacodynamics? Anyone? In simple words, if I would say, so pharmacokinetics is that what drug does to the what body does to the drug. So that is the process ADME. A is absorption, D is distribution, M is metabolism, and E is excretion. So first, the drug, when it is administered, it gets absorbed, then it gets distributed, it also gets metabolized, because like the food we eat and anything which we eat, it also gets metabolized in the body. Same way the drug also gets metabolized in the body. They follow some reactions which and those reactions are known as a biotransformation reaction, means the drug gets biotransformed in the body. And then and then after that, after the metabolism, generally the metabolism of the drug majorly takes place in liver, and also it takes place in kidney, but majorly the drug get metabolized in liver, and then it leads to the formation of either active metabolites or inactive metabolites. So there are active metabolites. So generally certain active metabolites are there, which are those active metabolites are only responsible for the drug action that happens in the case of prodrugs. And then there are certain active metabolites which are the reason for the adverse effect of the drug. And then there are inactive metabolites. So inactive metabolites means they excreted out from the body. Okay, they do not participate, they are not available for any action, they excreted out from the body. And through kidneys, majorly via urine, the drug gets excreted out. Then some amount of drug also gets excreted out through exhaled air and sweat and sweat. So these are the other forms of excretion, other mode of excretion. So pharmacokinetics, mainly it consists of 4 processes, that is absorption, distribution, metabolism, and excretion. Now there is one more thing which we want to understand in the pharmacological process of a drug, that is ROA. ROA means route of administration. So route of administration means that how we can administer a drug to the body. So now there are 2 routes by which the drug is administered. First is the topical, the systemic route, and the other one is the systemic route and the local route. Okay, so systemic route means when the drug is going in the body, like it is entering into the blood circulation. And local route means the drug is, wherever the drug is applied, the drug will have the action over that particular site only, which generally happens in case of ophthalmic preparations, optic preparations, ointments, paste. Suppose if you are burned and you are applying the ointment over that area only, so the action of that drug will be in that area only. A very minimum amount of drug get absorbed in the body. So that is the local route. And in local route, generally the drug is administered via topical route, that is, it is applied on in the form of jellies, paste, ointments, okay, and ear drops, eye drops, okay. So these are the local form, these are the formulations which are which are approved for the, which is having the local action, means or that the area only where the drug is applied, okay, or where the drug is administered. Then comes the systemic route. Systemic route means when the drug enters in the body and via blood circulation, it moves throughout the body, and wherever the receptor of the drug is present, so they bind there and then they will show its action. Now the systemic route also, it is of two types, that is oral and parenteral. Okay, oral route, that when we are taking the drug through our mouth and which goes into our gastrointestinal tract, generally the solid dosage forms and liquid dosage forms are taken by that route. That is, solid dosage forms means the tablets, capsules, okay, and syrups. These are the solid and liquid dosage forms. These are administered via oral route. Then we have the parenteral route. Parenteral route means when the drug is injected directly into the blood, so that is intravenous route. Intramuscular route, when the drug is not injected into the veins, it is injected into the muscles, but it takes very less time to get absorbed. Then intradermal injection, subcutaneous injection, intraperitoneal injections. So when any drug is injected, so that is known as parenteral route. Okay, so these are various routes by which the drug can be administered. Now, depending upon then in what formulation we have a drug, so accordingly the route of administration is decided. Like, we cannot inject a tablet. So if it is, the drug is available in the form of any tablet, it is to be taken orally only. Okay, we cannot inject any tablet. In case if it is available in the parenteral solution, then we have to inject it. We cannot drink it. Okay, so depending upon that in what formulation the drug is available, according to that, the route of that drug administration is decided. Now, in case of when the absorption of the drug, so now in case of when the drug is injected, like if it is an intravenous injection, then in that case the absorption route is bypassed. The absorption is not occurred. The drug directly enters into the body, into the blood circulation. But when the drug is taken orally, that is via in the form of tablet or in the form of capsule, now it first goes into your gastrointestinal tract. It goes into your stomach. Suppose it is in the form of tablet, it gets disintegrated, it gets diluted, and then after that it gets absorbed, and then it will be available in the blood circulation. So generally, when we take the drug orally, so in that case, the onset of action is delayed. Okay, and the drug also get lost through via first pass metabolism also, when majorly the drug get metabolized in liver, and it gets metabolized, and now it is no longer available to show its action, and only very small amount of drug is then available to show its action. And generally, when the drugs undergoes like first pass metabolism, that is also the reason the drugs are toxic to the liver. For example, is paracetamol, because paracetamol exhibits a high first pass metabolism, and the metabolite which is formed, it is very toxic to liver. So in case if anyone who has a habit of taking paracetamol, or it takes paracetamol very frequently, then in that case it leads some damage to the liver or liver impairment. Okay, so that is an example of first pass metabolism. Okay, so this is how the drug gets absorbed. Now the drug gets distributed. So the drug gets distributed via blood. Now sometimes the drug get bound with the plasma protein also, which is known as bound drug. The drug which is not bound with the plasma protein, that is known as unbound drug. And the unbound drug, when it reaches to the receptor, it binds with the receptor. Now when it binds to the receptor, it forms a drug receptor complex, and as the drug receptor complex is formed, now it will show its action. So that is the process of pharmacodynamics, which is known as simply that how the drug, or like what is the effect of the drug, or how what drug does to the body, means which is mostly concerned with the explanation of mechanism of action. What is the MOA or mechanism of action of the drug? Okay, then after that the drug also gets metabolized. Majorly the drugs get metabolized in liver, and then the drug also get excreted out from the body. So this is the whole process of ADME. And to understand this process is very important. So now coming back to the preclinical studies. So as we all know that preclinical studies are conducted on animals. Okay, and these are conducted on species, rodent and non-rodent species. So rodent species are the rat, mice. Okay, and non-rodent species are like the higher animals like cat, dog, monkeys. So on that also the studies are conducted. So now we need to study what is the pharmacokinetic of the drug, because from that only we'll get to know that how the drug is absorbing, how much time it is taking to absorb, then how the drug is getting distributed, how it is getting metabolized, what are the metabolites which are being formed, whether the metabolites are toxic or non-toxic, how the drug is getting excreted out of from the body, does the excretion, does the metabolites when they are excreting out, they are having some nephrotoxic effects, means they are nephrotoxic, they are toxic to the kidneys, or when during the process of metabolism, are they toxic to the liver. So from this we can understand that what from the pharmacokinetic process we can understand that what can be the adverse effects of the drug if it is taken, if it is not taken in a required dose, or if it is administered to any liver impaired patient or any kidney impaired patient, or what is the effect, what can be the, or how it affects the normal, how it can affect the normal body functioning of the body, of normal body functioning. So with the help of knowing about the absorption, distribution, metabolism and excretion, we, thus researchers, they can anticipate certain adverse effects which are related to the, or which are associated with the use of the drug. So that's why the study of pharmacokinetics, and also to understand that what is the mechanism of action of the drug, how the drug is binding with the receptor, whether the drug is binding specifically and selectively with that receptor only. So that is also required to understand that what is the effect of the drug when it is administered to the body. The next is proof of concept. Proof of concept means when you want to prove any concept, like initially when there is no practical which is done, any study is not conducted. So initially it was just a hypothesis. Okay, and then when you conduct a study, and then when you have a data to prove it, so that is known as the proof of concept. So when you conduct a study or a practical to prove any hypothesis, so that is known as proof of concept. So initially when we have discovered a drug, we have done the studies only like a kind of a assay development we have done, or in vitro the studies are done, but now we are actually administering the drug to the living organism, and with that we can understand that what is the actual effect of the drug. So that is known as proof of concept. Then drug delivery means that how the drug can be delivered, means what can be the route of administration depending upon what is the formulation. Now formulation optimization and bioavailability. So we also need to understand what is the bioavailability of the drug, because on the basis of the bioavailability only we can determine that what will be the till how much duration the drug action will be there in the body, or what will be the actual action of the drug, how effective the drug is. The drug can be so that is so bioavailability of the drug is studied, and formulation, in which formulation the drug can be administered, what is the what is the route by which the drug can be administered, whether the drug can be administered via two routes, like if it is available in two formulation. Sometimes the drug is available in injectable form, also in the tablet form. Like in injectable form, generally the drug is administered in emergency condition when onset of drug is required fast, and in normal condition we can give the same drug via oral route. Okay, and depending upon the patient condition, also severity of the condition, severity of the disease, and like so that that depends. So that formulation optimization and bioavailability studies are done. Then dose range finding. So dose range finding means we have to find the optimum dose or optimal dose which we can also consider for the human population as well, like the dose which we can also administer to the human. So dose range finding, generally these studies are done as initially it starts with a minimum dose like of 50 mg. Okay, then gradually the dose is increased, like from 50 to 70 mg, from 70 to 100, from 100 to 150, then to 200, and then to 250. Suppose at 250 mg the adverse effects of the drug is being seen, So between 50 to 200 mg, we have to find a optimum dose where the action of the drug is maximum. Okay, so that is known as, so in this that is dose range finding, or which is also known as the therapeutic index or therapeutic window of a drug. In between those two ranges only, the drug will have the maximum efficacy and lesser side effects. Okay, then after, when the preclinical studies are conducted, is completed, then after that the IND is filed. IND is known as investigational new drug application. Now there are various types of INDs, emergency INDs, commercial use INDs, so that we will discuss in the module number 5, when we will discuss about in detail about IND. And then you have then there are, what are the documents which are required along with the IND application form, so that also we will discuss. So then IND application is filed to the regulatory authorities along with the required documents, and generally it takes about 30 days time for the regulatory authorities to respond. In case if in those 30 days time the regulatory authorities does not respond, they does not object the IND, then in that case the IND become effective, the clinical trial process can start. Okay, but in case in between, if there are certain issues which occurs during the inspection process, auditing, or from the DSUR reports, if it is detected that the drug is not having side effects or adverse effects which are very frequent and serious, or maybe the drug is not effective, it is not as effective as it was in during the preclinical studies, it is not as effective in humans, in that case the regulatory authority can take some action. They can stop the clinical trial, they can pause the clinical trial, and after that query is resolved, and then after that the clinical trial can be resumed. So after preclinical studies, means once the preclinical studies are completed, IND application is filed. IND application is filed when the IND is approved, then the clinical trial on humans started. Okay, so now next is clinical development, or once the preclinical studies are completed, the drug enters into the clinical phase, or the clinical trials are conducted. So clinical trials are conducted on healthy volunteers in phase 1, okay, or the patients, depending upon whatever the condition is, okay, or whatever type of disease is. So accordingly the population is selected. Then the phase 2 and phase 3, the study is done on the patient population. Okay, and in phase 1 there will be very less participants. In phase 1 we generally study about the dose, like what can be the dose which can be administered to humans. Sometimes it requires a little higher dose than what which is administered to the animals, to humans. And then the dose tolerability, what is the mechanism of what is the actual mechanism of the drugs, or pharmacokinetics or pharmacodynamics in the humans. So for that purpose also the healthy volunteers are required, but that is in very less amount. The population is very less. Then after that the population gradually increases in phase 2 and then in phase 3. And phase 2 and phase 3 studies are conducted generally to get more knowledge about the safety and efficacy of the drug. Okay, so here in the phase 2 and phase 3 studies, the patient population is only considered. Okay, so in the clinical studies, we study about the safety and efficacy of the drug in human, and what pharmacokinetic analysis is done. Okay, and dose escalation, single and multiple dose studies, means we increase the dose, that what is the effect of the drug if the dose is increased. Sometimes the dose which is required for human population, it is slightly higher than from the animal. What is the effect of the drug when the drug is studied, or drug is given in multiple doses? So that is all studied in the clinical trial process. And bioanalytical method and development and validation. So bioanalytical methods, bioequivalence studies, bioavailability studies are conducted during the clinical trial process. For that is also conducted to see that how the drug is excreting out from the body, what is the effect of the drug on kidney when the drug is excreted out. Okay, so all these, like modeling, compartment modeling, and also that is all studied in the clinical trial process. Then once the clinical trial process on humans are completed, now the drug is ready to enter into the phase, the drug is now ready to enter into the market. But before that, again, the applicant, the sponsor, they have to file an application which is known as NDA, ANDA, or BLA application. Now, NDA, now depending upon that, what type of drug is, depending on that, the application varies. That is for NDA. NDA stands for new drug application, or if any drug is a new chemical entity, entirely a new chemical entity, or there is any drug, for any drug, if it is now reintroduced in the market, but it is already in the market, but now it is to be reintroduced in the market because it can now be used for some other indication, or or the route of administration of the drug is changed, or the formulation is changed. So for all those type of drugs, the new drug application is filed. Then ANDA is abbreviated new drug application, and abbreviated new drug application. So they are now, initially the drug, when they are discovered, they have a patent, they are marketed under a brand name. Okay, now when their patent expires after 10 to 15 years, now the drug can be manufactured, or they can be marketed by the generic name also. So to see the bioequivalence of the drug means to see that whether the effect of the drug is same as that of the branded drug in comparison to the generic drug. So for that, the ANDA application is filed, that is abbreviated new drug application. Then we have BLA. BLA is known as biological licensing application, and biological licensing application is filed for biological products like vaccines, serums, okay, human blood and blood components, cytokines, hormones. So any of these drugs, if it needs any of these components, if needs to be marketed, so for that, BLA, that is biological licensing application, is filed. Okay, then after that, the drug, again, it undergoes the FDA, the regulatory authority approves it. Generally takes, there are a lot of meetings which are conducted, inspections conducted. It is a long process. It takes at least 6 to 10 months of time for the drug to for getting approved for marketing. Then after that, the FDA approval, or the drug, or the concerned regulatory authority approval takes place, and the dossier is also submitted, that is the drug dossier is submitted, and the drug is registered. And then after that, drug is registered with the regulatory authority, and then after that, the drug is available in the market, which is known as, and the process is known as post-market monitoring. Okay, now once the drug is available in the market, then the phase 4 studies, or known as post-marketing monitoring or post-marketing surveillance or pharmacovigilance studies starts. So in that we so in that, the marketing authorization holder, they plan certain pharmacovigilance activities, and by which they get the knowledge about the adverse drug reactions related to the drug. Okay, because their clinical trial process, they are conducted in a very controlled settings, close monitoring of the participants takes place, and when the drug actually enters into the market, it is subjected to a larger population, and sometimes the drug is used off label, off prescription, which may lead to some kind of adverse effect. The drug can be administered to any population to which it should not be, like for example to the pregnant or lactating woman, or to any hepatic or renal impaired patient, or in case of any overdose occurs, in cases of medication errors occurs. So now this is pharmacovigilance is entirely a different field. Okay, so which comes, and the pharmacovigilance activities, they are planned by the marketing authorization holder, which are conducted by the marketing authorization by the pharmacovigilance professionals. There are various surveillance, like active surveillance, passive surveillance is there, by which they come to know about the adverse effects related to the drug, and also not only the adverse effect, but also they'll come to know if the drug can be used in some other indication. Okay, in some other indication or in some new indication, then that also. So overall risk benefit analysis is done during these activities, and the report, annually the report is submitted to the regulatory authority, which is known as PBRER or PSUR, which is submitted to the regulatory authority, so that the regulatory authorities will also come to know about the drug, its effectiveness, its benefit, the risk and benefit which is associated. So that all is done during the post-market monitoring. And for reporting that this adverse drug reactions, there is a system which is developed by the regulatory authorities. Like for FDA, there is FDA adverse event reporting system. In European Union, there is a yellow card scheme. And then in India also we have an adverse drug reaction reporting system, which is governed by IPC, Indian Pharmacopoeia Commission, which gives you a form which can be filled up by the doctors, or by the doctors or other healthcare professionals, or itself by the patient itself, and then a proper action is taken on that. So what is pharmacovigilance in brief, that we will discuss in module number either it is module number 8 or module number 9. So just for instance, you can understand, then once the drug enters into the market, the pharmacovigilance studies or post-marketing studies starts, and it lasts till the life cycle of the drug. So this is the whole process of drug development. Okay, understood? Is it clear? So now we'll quickly take it up, that is discovery. New medications are typically found by researchers through new understanding of disease mechanism that enable researchers to create a treatment to halt or reverse its symptoms. Now, whenever the discovery is done, now the process is known as rational drug discovery, in which first the researchers, they try to understand about the disease, its mechanism, how the disease is progressing, what is the molecular mechanism behind the development of the disease. And then on the basis of that information, they try to create a drug or a treatment which can reverse the symptoms of the drug. Then molecular compounds undergo several studies to determine any potential therapeutic benefits against a wide range of diseases. So now there are various molecular compounds, chemical compounds, maybe from whatever source they are. Then after that, there is a huge chemical library or a database from which these molecules are being screened out. They are subjected to various computational methods, and from that only we'll come up with one lead compound which can be considered as a drug, if it is which can be considered as a drug, as it is safe and effective to use. Then treatment now in use that have unexpected consequences. Now there are certain treatments which are available, which are having some unexpected consequences, so those treatments need to be stopped. Some drugs, they are toxic, but they are effective, but they are also toxic. They are having serious adverse effects, so we cannot move forward with those drugs. So there are various combination and combinations are done, and then after that a drug is discovered. Then new technologies, like those that offer new techniques to change genetic material or tailor medical items to specific body parts. So there is a new technique which is developed, which is still under study, that is known as pharmacogenomics, in this technology it offers it is an advantage that the personalized medicines can be created. With this technique, the personalized drugs or personalized medicines can be created, in which the drug is made according to the genetic makeup of, or according to the genome of a particular person. But of course, that will be a very costly therapy. So that is a process known as pharmacogenomics. So this is a new advancement which is now being done, or there are new there are new tests and development in this, or experiments are being, researchers are still being done. So thousands of chemical may be candidates for development as a medicinal therapy. At this point in the process, only a limited number of molecules meanwhile appear promising and warrant additional research after preliminary testing. So this process is not easy as it sounds. It is a very, very complex process, which is also time taking. Lot of manual and computational expertise is required. Manual work is also required. Computational expertise is also required, and it requires a lot of financial expenditure also to develop, to discover a drug, and then to make that drug suitable for commercial use. When a potential molecule is chosen, then after the discovery, the next process starts, that is development. So when a potential molecule is chosen for development, researchers conduct tests to learn more about how it is metabolized, distributed, and eliminated. Means the pharmacokinetics of the drug is studied, its prospective advantages and method of operation. Method of operation, I guess, is not the right word. It should be route of administration, that how the drug can be administered, what is the ideal dose or what is the optimum dose at which the drug is having the maximum bioavailability, and at which the drug is showing the lesser side effects. The ideal dose for the medication, such as that what is the doses form in which the drug can be administered, means the formulation. And according to the formulation only, it can be decided the drug either it has to be taken parenterally or orally. Then toxicity is the term for unfavorable reaction or side effect. So the drug is said to be toxic, or the toxicity means the adverse effects which occurs or adverse event which happens with the use of the drug. How it differs depending on the gender, race, or ethnicity of the group of people. Now, the effect of the drug can also depend upon, or can also differs on the basis of the demographic characteristics, depending on the race or the ethnicity. How it interacts with other medications and therapies, like which is drug-drug interaction, drug and food interaction, that is also studied. Its effectiveness in comparison to equivalent medication. So if in the market already for that disease other alternative treatments or medicines are available, then how effective the drug is in comparison to those alternative treatments. Okay. Now we will study about the preclinical research. So mostly about preclinical research we have already discussed. We'll just we'll revise it again. So first, the drug discovery process, that is which consists of 4 stages. That is drug discovery, where we find all the target discovery, when we find all the targets which can be responsible for the development of the disease. Then target validation, where we eliminate the wrong target, or we confirm that this target is actually responsible for the development of the disease. Then lead generation and refinement. The process is also known as lead identification and lead optimization, in which the molecule is generated which is having the maximum efficacy and minimum toxicity. And then after that, it is subjected to preclinical development when the drug is tested on animals. Now, preclinical trials test the new drugs on non-human subjects for efficacy, toxicity, and pharmacokinetic information. These trials are conducted by scientists in vitro and in vivo with unrestricted doses. 2 types of preclinical research are there: in vivo, which is conducted in the human, in the body, in the living organism; in vitro, which is conducted in the laboratory. Okay, so there are 2 types of studies which are generally conducted during preclinical testing. That is the pharmacological studies, that is to study about the pharmacokinetics and pharmacodynamics of the drug. And the next one is the toxicological studies. So there are various toxicity studies conducted, like for example reproductive toxicity studies. Then you have mutagenicity studies, carcinogenicity studies, immunotoxicity studies. So all these studies are conducted to get knowledge about that on a specific organ, or specifically the drug is toxic in that way or not, which otherwise can be the serious toxicity, which can be the life-threatening toxicity, or which may lead to some permanent abnormality, or which can lead to death of the patient as well. Then FDA requires researchers to use good laboratory practice, GLP, defined in medical product development regulations for preclinical laboratory studies. Now, the preclinical studies, they are generally conducted in the laboratory, and for conducting the studies in the laboratory, GLP guidelines, known as good clinical laboratory practice guidelines, are to be followed. Now, what are these good laboratory practice guidelines? So these are the GLP regulations that are found in 21 CFR Part 58.1, good laboratory practice for non-clinical laboratory studies. These regulations set the basic requirement for that how to conduct the studies, how will be the who are the personnels which are involved in the studies, what is their qualification, training. They should be well trained in GLP. They should know that whatever task is assigned to them, how they have to perform that task. Then what about the facilities, that what are the facilities should be present? There should be a proper storage condition. There should be a proper room for the animals. Okay, proper clean room for the animals, air conditioned, whatever the requirement is, that should be fulfilled. Then there should be a separate room for conducting different kind of studies. Then all the equipment should be well calibrated. A computerized system should be validated. Okay, and there should be a written protocol for conducting each kind of study, like there should be the SOPs for each and every step which is to be conducted, and there should be a study plan that study plan, according to that study plan which is made by the study director, and that is only, and according to that study plan only the study will be conducted. Operating procedure. So as I've told you that for performing each and every task, there should be a standard operating procedure. Study reports are to be made, means once the preclinical studies are conducted, there should be the study reports, and the study should be, whatever the results are obtained, that should be reported in those study reports then, and a system of quality assurance oversight for each study to help assure the safety of FDA regulated product. Means there should be a proper quality assurance programs in which the quality assurance personnel, they are responsible for conducting inspection and audits in the when the study is in process, so that they can come to know that the study is conducted as per the study protocol only, or the study or according to the study plan only. And in case if there is any shortcoming or any finding, so that should be resolved, or proper audit report is made, and that should be submitted or presented during the time of FDA inspection, if it happens in the preclinical laboratory or in the facility where the preclinical testing is being conducted. Okay, so preclinical investigations are typically not very big. These studies must, however, offer thorough information on doses and toxicity levels. Researchers evaluate the result of preclinical testing and determine whether human testing for the medicine is necessary or not. Means if the drug is having the promising effect during the preclinical testing, it is having the efficacy and safety, then the drug will proceed further to conduct the testing on humans. Otherwise, no human clinical trials will happen. So evaluation of the result which is obtained from the preclinical testing is very important, and the same information is submitted to the regulatory authorities as well, so that the result of so that they can grant permission to conduct the clinical trial on humans.